And if you go down the checkbox, you can see that if I were studying for this test, especially in a study group, by the way, and you got 2 more days this week, you got Saturday school. If you need me, I'm here from 8 until 11, and then to 10, and then from 11 to 12. Monday's your test, but then you have your final. This is going to be on the final as well. So it's going to be important that you get it. I would have, by this point, you've had this for about 2 weeks now, emailed to you and printed for you and handed to you. So there's absolutely no reason why I shouldn't be able to, if I wanted to, tell you, let me see your index cards with your vocabulary key terms. Let me see them with all these key terms that you should know. No one in this room should be surprised about any word that's listed here. My guess is a lot of you do not have these key terms defined. Even though it was a homework assignment, even though the chapter work's already been turned in, even though you have practice test questions in the back of the chapter, my guess is it's not done. Okay. Here's some key concepts. Where is Mendoza getting these questions? I'm going to say it one more time. The back of the chapter. What chapter is that? Chapter 8. And these key terms, this study guide. There's check boxes. Why did I give you a checkbox? Why is there a checkbox involved? Because you need to check off that you can do this. Every one of these that you don't check off is probably a wrong answer on a test question. Does that make sense? Key terms, do you understand them? My guess is, again, some of you don't even know what the Krebs cycle is. What is lactic acid? Some of you know. My guess is a lot of you don't. You should know. So you need to check these off one at a time, and as you check them off, how do you know that you know them? That's where study group comes in. That's where you can say, hey, do you know what ethanol is? Where's ethanol made? Where does ethanol come into this? Oh, ethanol is in fermentation. It's in alcohol fermentation. Oh yeah, that's right. That's what happens when there's no oxygen available. Okay, cool. That conversation should be happening in a study group. If not, if you insist on doing it on your own, you should be looking it up. Where's ethanol? Where's glycolysis happening? What are grana? What's a hydrogen ion acceptor? What's cyclic phosphorylation? Ooh, it's a fancy term. What's the universal role of ATP in metabolism? What's describe the structure of ATP? You don't have to memorize the structure, just describe it. Describe it in the sense of the things that I've talked about in class for about 3 weeks now. So things like nitrogenous base, adenosine, and 3 phosphates. Just something that simple would be all it would take for you to be able to get that question right and to check it off your list. Did you know that? My guess is a lot of you didn't. Should you have known that? Absolutely. Did we cover it in class? Sure did, many times. Is it in your reading, chapter reading? Yes, it is. Is it in the study guide that should have been submitted Monday or now Tuesday? Sure is. All right, then it goes on. And then we go through cellular respiration. Well, what are the steps to cellular respiration? That was today's bell work. Where there it is, glycolysis, Krebs cycle, electron transport chain. Wow, that would have been easy. Highlighted in dark black, the 5th thing that you should know in something that was given you two weeks ago. Identify glucose as the primary as respiratory substrate. Explain the use of respiratory quotient. What's RQ? Well, we're going to look into that. You should know that because you should have read it, should have did the problem. It's a ratio. It's not really that big a deal. But if you haven't done the work, you don't know what it is. All right. What's chemiosmosis? So there's that word osmosis. What's chemoosmosis? What's fermentation? What's a hydrogen ion acceptor? And then you keep going for photosynthesis. So those are the kinds of things that you would expect that people would go. And you can go, if you needed more work, you can go to the link here, or you can go here to a periodical. You know, they have some listings, some references for you to look up if you absolutely need to. Do you need to do that? No. Why do you need to do that? We literally are going through it in class. But whatever, if you feel more comfortable with that, go ahead and read it. Do whatever you need to do to on this test. Adenosine triphosphate, the structure of adenosine triphosphate, one of the first things in that list of things to do.
Miriam here.
In the back. Well, there it is. I do a simpler structure in class in the previously posted video, right? You have ribose. It's just a sugar, 5-sided sugar. We've talked about it. You studied it. You tested on it. You're going to see it on the test, the different glucose and ribose and fructose. Those three were the 3 sugars I said you needed to know. I said it months ago. I said you're going to see it on the final, and you are. But this is just a model that just shows you the molecule as a whole. You don't need to be able to draw this, right? But you know you have ribose, you have 3 phosphate groups, and you have adenine. Where is ATP used? Where is ATP used for metabolism? Well, we've talked about this. Endocytosis, exocytosis, where you're changing the entire membrane, changing the endoskeleton. It's going to require, or one form of endocytosis is phagocytosis, right? You've been tested on this already. So this is going to require a lot of energy to be able to change your whole cell just to take in this giant piece of whatever it is. So it's going to take you effort. It's going to take energy. What form of energy is used by the cell? ATP. Any work that's done is going to require ATP. Mitosis, cell division. When you're moving things around, you're moving. What do you need? Energy. What energy does a cell use? ATP. Hydrolysis of ATP means you're going from ATP to ADP. Is that clear? When you see hydrolysis of ATP, you're going from ATP to ADP. This is a flagellum. It's moving. It's a flagellum in a bacteria. It's moving. This pylori, this Helicobacter pylori, causes ulcers in the stomach. This thing swirls around its tail. What's the only way it's going to be able to move its tail around? ATP. This is a bacteria. It doesn't even have a nucleus, but it has ATP. Does not have mitochondria, but it has ATP. The basic unit of energy for living creatures on this planet. Maintenance of your body temperature. If you want to keep yourself warm in the cold, you shiver. What does shivering do? How do you shiver? How do you contract those muscles? ATP. So, very basic concept here, right? Here's a mitochondria. This is what a mitochondria actually looks like. We draw a very, very simple drawing, but when we look under an electron microscope, this is the kind of thing you see. You see the outer membrane of the mitochondria. There's an inner membrane, so it's a double membrane structure. You have these folds that form a matrix, right? So there's all kinds of spaces in here. What do we find? We find all these different enzymes in there. Here's where the Krebs cycle is happening, right here in this area. Do you need to know this? No, this is just for reference. This is just to give you an idea about when you see a drawing like this. This is what the drawing we have. This drawing is based on this microscopic picture. This is the picture that we got using technology. This is the artistic drawing of it. That's all. It's an easy page, isn't it? All of 2 minutes to look at. And you could read the details of it, right? It says here, amine oxidases on the outer membrane surface, phosphorylases between inner and outer membranes. There's an inner and outer membrane here. ATPases on the inner membranes, so there's ATPases here, right? And soluble enzymes for the Krebs cycle are found here. So that's how we know where these processes are going on. This is ATP, 3 phosphates on adenosine. And of course, there's a ribose sugar in there as well, right? 3 parts to a nucleotide. Don't forget that. Adenosine is the nitrogenous base, then the ribose sugar, and then to the ribose sugars attached 3 phosphates. Yes? For every phosphate that leaves, you get this much energy, 30.7 kilojoules of energy. Do you need to know what kilojoules are? No. You should by the time you're in physics, but let's not worry about that. What I need you to know is that every phosphate is going to give you some energy. Every time you take a phosphate out, it gives you an energy. So ATP going to ADP gives you this much energy. Take ADP to AMP gives you a little more energy. Less than 30, but a little more. Yes? We got it? So if you build it, what are you doing? Remember the first law of thermodynamics. You have to conserve energy. So if I make an ATP, I have to put at least this much energy in, right? Because that's what I get out when I take it off. So that must have been what I put into it. At least that much. Turns out you have to put in a lot more to get this much in the bond, right? Because it's inefficient. You always lose a lot of energy to heat. We've discussed that already, yes? Okay, so there's going to be a lot lost to heat. But at least 30 joules of energy, 30.7 joules of energy, must have been put into these bonds. Where did that energy come from? Well, it depends. It depends on which process are we talking about, photosynthesis or respiration. All right, so inorganic phosphate, where does phosphate come from? It comes from rocks. So phosphate is a mineral. It's found in rocks. So ADP is a necessary substrate. Add a phosphate, you make ATP. Pretty simple, pretty straightforward. That's the process of ATP, which is why it surprised me that as we were going through the last video, people were like, what's ATP? I don't why? You should have read this, right? You should have already read. We discussed it already earlier in the year. Here's some photosynthetic changes that happen. This is kind of a really fantastic outline of what's going on. So you really gives you a headache to read all the details of all the enzymes. Well, my God, they give you here a summary of it. There it is. It's summarized for you. The entire chapter 8 is summarized right here in this one diagram. If you learn this, you probably get 5, 6 questions right on the test. I mean, that's it. Boom. Carbon dioxide and water go into photosynthesis. Out of photosynthesis comes glucose. Of course, you need light going in because that's your source of energy. Are there a lot of other little steps? Yes. Should you know that you're using a concentration gradient to turn ATPases to make ATP so that you can take the ATP to the Calvin cycle, that you have light reactions and then you have the Calvin cycle, you have photosystem 2, then photosystem 1, that you're taking water, you're breaking it up to get the electrons. Yeah, you should know all that. That's all part of photosynthesis. But a summary is carbon dioxide and water go in, glucose comes out. That's easy. You should know that. Yeah, I can guarantee you're going to get at least 2 questions on a test like that. They have to do with just that. You'll also get a question on a test that asks you what's water used for. In some way on the test, whether you know what water is used for in photosynthesis. What does water get turned into in photosynthesis?
You told us this yesterday.
I did. I told it to you like many times. I told you 3 weeks ago. I told you yesterday. It's in the reading. What's water? And what do we get out of it? What is photosynthesis? Not we. What do plants do with the water? They break it up. Very first step in photosynthesis, you break it up. And what do you do with the oxygen? What do they do with the oxygen?
They produce that waste.
They release it as waste. They don't need, the photosynthesis reactions don't need it. Notice that what they do in their respiration, in their mitochondria, look at what they're doing. What are they using?
Oxygen.
So they use oxygen, they just use it in a different place, right? They make more oxygen than they need. So they are producing more, they are releasing a lot more oxygen. So photosynthesis, already you know a lot of questions. There's a lot of questions that just got answered. What's a question I might ask in a test? Where did the oxygen in photosynthesis come from? Where it came from water. What step? Photosystem 2. How? Light was used to break it up. The energy from light was used to break it up, right? Is that simple? The electron in water, the electrons get pulled into the electron transport chain here in photosynthesis, right? And the hydrogen is used for the hydrogen ion concentration. But those are details. The summary here is pretty good. Notice here, what's being released?
Heat.
Every time. Heat has to be released. ATP provides energy in metabolic pathways. While some energy is stored in the chemical bonds, some is lost to heat. In fact, most is lost to heat because we're in it. There's no 100% efficient system that we know of. Yeah.
So I know it's lost to heat, but how does the heat get released?
It radiates. Have you ever worked out? Have you ever worked out with a bunch of guys playing basketball or girls playing basketball? Have you ever been near them?
Yeah, it's hot.
Don't they feel hot? That's the heat they're releasing. They release it. They radiate it. Just like a hot lamp radiates it, right? It leaves and goes into the air. And also by touching. So heat energy can be transferred by touching, by convection, which is that feeling of heat from a distance. So there's a lot of different, again, I wish we had time to go through the different methods of heat transfer. All right, cellular respiration is what goes on here. What do you need for cellular respiration? You need oxygen, and you need what? Fuel. Usually it's glucose, but it doesn't have to be glucose. It could be a protein. It could be a fatty acid. It could be a nucleotide. It could be a lot of things. But it's somehow, usually we always just stick with glucose because glucose shows you all the steps of cellular respiration. So let's go ahead and continue. You should be able to answer these, right? Should be able to answer these. Let's see if we can answer them quickly. What are the starting materials for photosynthesis?
Water.
And by the way, I'm not going to answer those other questions above. You guys can answer these. I think it should be straightforward. Yes, H2O. We start with water.
Light.
We have light. We need light, yeah. What else? Photosynthesis needs water and light. I didn't say the light reactions. I said the whole process of photosynthesis. Does photosynthesis need oxygen?
No, the oxygen comes from the water.
Interestingly, the oxygen comes from the water, and in fact, it is what? A waste product. So O2 gas is a waste product from photosynthesis. What else does photosynthesis use? Carbon dioxide. That's right, CO2 gas. And you could put NAD+, and you can put all kinds of other things, but bottom line, these are the things from outside the chloroplast that you're going to need to bring in if you're going to do photosynthesis, right? What are waste products that produce? Oxygen gas. What's the role of hydrogen carriers like NAD? Is there NAD in photosynthesis? No, it's NADP. What's the role of NADP in photosynthesis? Right, that's right. Transfer electrons and hydrogen ions to ETC and Calvin cycle. Right? Because the light reactions, from the light reactions, you transfer those electrons, those high energy electrons, to the Calvin cycle. Also, the NADP. Well, that's what NADPH does, is it transfers it to the Calvin cycle. NAD is used in plants, but not in photosynthesis. Role of ATP. Energy source. Say energy source for cells. And in fact, you can get a little more detailed and say used in coupling of reactions, right? So we can couple them to different reactions. We talked about that. You drew it, you colored it. We had it in the other video. Overall biological role of photosynthesis is what? Very good. And what it does, really, let's use a little more fancy words. You're right. Providing food for the plant. Okay, that's fair. But I'm going to get a little more detail if you don't mind. It's to transform light energy, that's right, good, into chemical energy. Specifically what? Glucose. And it's not just glucose. Glucose, proteins. It's used to make a lot of stuff. So we can say chemical energy and more complicated molecules. So what do we call this? What is this called? When we take simple molecules, simple to complex. What kind of metabolism? Somebody said it's anabolic. So it's an anabolic pathway, right? What does anabolic mean? Building, right? So photosynthesis is used to build. We build things, and when I say we, I mean life, not humans. We don't do that. Humans don't do this. Animals don't do this. This is done by photosynthetic bacteria, by plants, and by algae. This is not done by humans or any animal that I know of. All right, cellular respiration. Do you think it's going to be anabolic? Think about it. Do you think you're building something with cellular respiration? No, doing the opposite. So everything in cellular respiration is exactly the opposite. So I'm going to tell you, that's why I always start with photosynthesis, because that's where we're capturing the energy. And let me tell you that if you know one, you know the other, because the other's just going to be opposite. So starting materials is what for cellular respiration? AT is starting. What do you start with? What's the
Oxygen.
Right? We'll start with oxygen. Isn't that interesting? The waste product of photosynthesis is used by cellular respiration. That's interesting to me. Oxygen, what else do you start with in cellular respiration? You guys have to know this. I'm not going to tell you. What do you start with in cellular respiration? What's the first step? Right, glucose. Now I like that somebody said glycolysis. I love that somebody said glycolysis. Look, listen. Glycolysis. Glycolysis. What does glyco and then lysis? What does lysis mean? Lysis? Break up. Break up or break down. And what does glyco mean? What do you think it stands for? It's the first step so it's just that simple. The first step in cellular respiration is glycolysis, and it just means you start with glucose, right? So starting with glucose, that's the first step if you start at the earliest possible stage of cellular respiration. So we're going to go ahead and say glucose. Now that's interesting for me as well, because you see here, you have glucose over here. So the plant produces it, and cellular respiration uses it. Remember, cellular respiration happens in the plant as well. I keep saying that because people leave biology often with a misconception that plants don't do cellular respiration. They do. They use that sugar. All right. What's the waste product of cellular respiration? What's put out by cellular respiration? CO2. You know, and here's another clue for you when it comes to cellular respiration. You do cellular respiration. So if anybody sits there and tells me that you take in carbon dioxide, I'm really sad by that. I hope everyone knows that you're using oxygen and that you eat glucose, so you're using glucose. Everyone in here should know that. And since this is what you do, this is you, right? So it's interesting, yes, the CO2 is a waste product. And what else is a waste product of cellular respiration? Water. We actually make water. We're making water all the time. Again, very interesting, isn't it? The product, the waste of cellular respiration is what? Is used by what? Used by photosynthesis. And isn't that interesting? You learn a lot from filling out a table like this, don't you? Role of hydrogen in ADP, exactly the same. Except it doesn't take it to the Calvin cycle, it takes it to the ETC, right? Takes it to the electron transport chain. And this is NAD. NAD is what's used during cellular respiration in the mitochondria. It's transferring electrons. That's what they do. And hydrogen ions, right? That's what they're moving these things around. That's why they're called, and look, the clue is in the word, guys. The clue is in the word. The word is carrier. It's carrying something. What's it carrying? Well, it's not carrying a turkey. It's carrying electrons, and it's carrying hydrogen ions. The electrons have energy in them. That's where the energy is coming from, is when they make those bonds, those chemical bonds. Remember, chemical bonds are sharing of electrons, right? Yeah.
So would it matter if the energy is positive or negative?
No, but the electrons are negatively charged. So that's really where we're at there. But no, it's just energy being transferred. The only thing that the cell is concerned with is, can it do what it needs to do, right? It's not really concerned with anything. It's just, that's the way, that's what's gonna, either it works or it doesn't work. All right, well, what's next? The role of ATP. What does ATP do? Same thing, right? Same thing. It's used to run metabolism. Whether it's catabolism or anabolism, it doesn't matter. What's the overall biological role of cellular respiration? Is to do what? To break down complicated molecules and get energy from them to run the reactions of life. And specifically, these are what? It's a catabolic pathway. So, sorry. So there it is. That's that chart filled in. Took a few minutes. Should have taken you a few minutes, I think, and I think you can answer all these, right? No, but you may go to the office. Use the office pass. You guys can answer these accurately?
Yeah.
Okay. I heard, yeah. If you have problems with this, maybe you should see me on Saturday or during advisory tomorrow. All right? Because tomorrow, by the way, Friday is a formal Friday, so we don't have time for tutoring it tomorrow. We're going to be in the auditorium on Friday, rather. We only have tomorrow. Yeah. So 4. This is that RQ. It's just a ratio. It's just CO2 to O2. Whenever you're looking at a ratio, you have to think, it's going to give you a number. That number, this is, by the way, known as a quotient. That's why it's called RQ. A number that represents a ratio is called a quotient. All right? So here you have a respiratory quotient. It's a number that's going to represent respiration. And how do you measure respiration? Cellular respiration. Think. How could you measure respiration? How do you measure cellular respiration? These are questions that I'm going to ask on a test. So you should be, often you guys are telling, your parents will ask me, well, how do they know what to study? I tell them repeatedly. I tell them in class what the test is, what's going to be on the test.
Parents ask that?
Yeah, I've had parents ask that, yeah. And I literally have answered, I've answered exactly what I'm telling you. I'm telling you. Huh? How do you measure cellular respiration? So here's my question. Here, let me ask you a question I might ask on a test. You ready? Could I measure cellular respiration by measuring how much glucose I'm producing? Could I measure cellular respiration by measuring how much glucose I'm producing?
Is glucose produced the same?
No. So the answer is no, you cannot measure cellular respiration by how much glucose you're producing. Why? Because glucose is not produced during respiration. Could I measure cellular respiration by how much oxygen is produced? No. Why? Because oxygen is consumed. See how easy that is? So I think, what could I use to measure cellular respiration? How much what? How much what? How much NADH is produced? Okay, I could do that. It's hard. I'd have to isolate it. I'd have to stop the reaction periodically. It would be complicated. I could use NADH. How much NADH? I could use how much ATP is produced. But again, I'd have to do a lot of biochemistry, a lot of isolation.
Can you use water?
I could use how much water is produced. Yes, I could do that because you're producing water. For sure, that's a good one. But an easier one might be measuring how much carbon dioxide is produced. So if I'm, and of course how much oxygen is what? Consumed. The easiest one to measure, and you're absolutely right, I love that most kids don't even think about water, right? So it's excellent that you said water. How much water is produced is a good measurement, could be used as a measurement of how much cellular respiration you're doing. Yeah.
So they take in oxygen, but what if they take in too much? They just get rid of it right back?
What? The animal? You do it all the time. Why? Do you ever think, the question he asked is, you're taking in oxygen, what if you take in too much oxygen, you're not using it all? You never use up all the oxygen you take in. That breath, do you ever wonder why when someone's doing CPR, they breathe out of their mouths and into somebody else? How does that help them?
I've never thought of
How could they help them? You're breathing it out. What are you breathing out?
carbon dioxide and oxygen.
And oxygen. Because you don't always take in all the oxygen. You don't absorb all the oxygen. There's still a lot of oxygen left when you're breathing out. It's just you're breathing out a lot of carbon dioxide. Does that make sense? Mm-hmm. So it does help someone to breathe for them. It's not as good as them breathing on their own, but it's better than not breathing at all, right? So yes, they don't take it all. They just don't absorb it all into their blood, all right? So CO2 is produced, oxygen is consumed. If we take how much CO2 is produced and divide it by how much oxygen is consumed, we get a number. The higher the number, that means the more CO2 is produced, if I have a lot of oxygen consumed, is that going to make this number smaller or bigger? If this number is really big, smaller. It's going to make the quotient smaller. How much CO2 is produced, it's going to make it bigger. Does that make sense? Okay. So when we look at the RQ, how much cellular respiration is going, the respiration quotient, when we look at that number, a number that's bigger than 1, it carbohydrates some anaerobic respiration. So if this number is bigger than 1, if this number is bigger than 1, I'm producing a lot of CO2, but I'm not consuming a lot of oxygen, right? If it's bigger than 1, this number is bigger than this number. Does that make sense? So that means that I'm not using a lot of oxygen, but I'm still producing a lot of CO2. I'm probably doing some kind of anaerobic respiration. Hopefully you know what anaerobic respiration is. Who knows what that means, anaerobic respiration? In the case of humans or dogs or animals in general. Yeah? That's right, you don't use oxygen. What is that process called? Fermentation. Excellent, excellent, very good. Fermentation, that's correct. So this is when fermentation is going on. You expect to see a lot of CO2, but not a lot of oxygen. That's why the respiratory quotient is such a good number, because we're able to, it tells us something about, not just about cellular respiration, but also what's going on. Is it respiration? Is it fermentation? You know, what is being processed? Where's the ATP coming from? If you use carbohydrates like glucose, you're going to get a 1. You're going to produce as much CO2 as oxygen you use as much oxygen as CO2 as the CO2 you produce. They're going to be equal. So you know you're burning a carbohydrate at that point. If you feed this cricket protein, you're not going to get a 1. You're going to get a 0.9. What that means is that it's going to consume more oxygen than CO2 is produced because there's less carbon in proteins, right? There's 6 carbons in carbohydrates, but there's less carbon in a protein. Does that make sense? That it's able to make it a CO2. I'll show you why and how in a minute. Remember this. Remember these numbers when we look at the pathways in the next slide. Okay? Fat is going to give you a 0.7. Again, you're going to have a lot more oxygen consumed than CO2 you're producing. And carbohydrate associated with organic acid synthesis is really, really small. So you got a lot of oxygen consumed, not a lot of CO2 produced. So the numbers are really small. So at that point, I think this is just plotting and analyzing what's happening during germination, right? And what are you germinating? Seeds, right? So you can take seeds and grow them, and you can measure an RQ for this, how much CO2 is being consumed and how much oxygen is being produced, and you get numbers. Which one of these are you going to put on the x-axis? Are you going to put days of germination on the x-axis? Which of these is the independent variable? That's what goes on the x-axis. Which one of these depends on the other?
I can't see what that says.
Let me zoom in here for you. There you go. Which is the independent? Which is not going to depend on the other? Which one of these two?
The days.
This one, or the days are independent. It doesn't. The RQ value is not going to define the day of the week it is, or how many days have gone by. How many days have gone by will define the RQ. So this one impacts this one, so this one's independent, so that would go here. Dependent goes here. So 2 and 0.65, 4 and 0.35, right? You guys can do that. I expect to see, when I grade these, I expect to see that done well, yes? And I should see 2 lines, one for seedling A and one for seedling B. Fair? I don't think that's something hard.
What if we already typed it in? Can we redo it in like?
Yeah, always. But remember the 14th, December 14th is the last day. All right, so that seems easy. So by the way, we could use this information to determine what's being eaten, couldn't we? If we took and we said, well, we got this RQ value, right? And it's about 1. What did that person eat? There's the quotient. There's the RQ, right? Respiratory quotient. Here's the 1, right? Isn't it 1 mostly? There was something happened. It was 0.7. What was digested when we measured at 0.7? We had a fat. There's some fat eaten there. And here's something that was 0.9. What was that?
Protein.
There was some protein. So whatever this thing ate, there was some fat, some protein. There's some, and that's about this is probably protein as well. But then what happened? Mostly what? Mostly carbohydrates. That's easy to read, right? That's easy. That's life. It's easy. Here's a chart. And I think you should be able to answer all those using those tables. Being able to read these tables and charts are very important for your future. You have to be able to read graphs and tables. If you can't, you're going to be in trouble. I don't care what you want to do after high school. You have to be able to read charts and tables. Even if you don't understand the basic nature of it, you should be able to tell the relationship between RQ and the amount of, and the material being eaten. So here is a graph. You were supposed to graph this. Hopefully you did that. Here is one. This is either going to go on the X or Y. These two are either going to go on the X or the Y. Now, the X is the independent variable. Which of these two, the RQ or the days of germination, is the independent variable? The days of generation after generation is the independent variable. This does not. The RQ is not going to determine how many days, but the number of days will determine the RQ. Yeah. Yes, you may. So this is going to go on the X. So you have to take 10, divide it by however many divisions you have here. That's how much each one is worth. Then you have RQ on the Y then. And you're going to have two lines, aren't you? A and B. Now some people, I saw this, they graphed bars. They said day 2, they have this bar and this bar. Day 4, they have this bar and this bar. Yes, that's all right, but you're not going to get as much information as you will from a line graph. Also, as a clue that they wanted a line graph, how did they graph it up here? Like a line graph. So some people just force something because they just have a vision in their mind. They're used to doing bar graphs. It's easier. I'm going to do a bar graph. But a line graph is going to give you more information. Sometimes. Sometimes a bar graph is the one you want to use. So make sure you're using in this case it would be the line graph that you'd use, and you put the X here, the Y here, and graph it, and then you can answer the questions and interpret the data. Okay? Get that done. I'm interested to see what you have to say. Recording. So, just to be clear, when we're looking at this graph here, at this table, excuse me, you're telling me that the RQ value, sorry, wrong again. The RQ value between these two is what? 0.7. Everybody sees that the RQ value here is 1, right? Does that make sense? The RQ value here is 1. What about this one? What's the RQ value here? Now, at 30 degrees, it's warmer, right?
That's Celsius.
This is Celsius, so it's much warmer. 10 degrees Celsius is a much bigger deal than 10 degrees Fahrenheit. Right? Yes? All right. So at 30 degrees, now what's the RQ value here? What is it? 3.57 divided by 5.12 is still 0.7? Okay. So 0.7. So what is the cricket eating initially, the cricket was eating what? Carbohydrates, right? How did we find out? How do we know that? Because the RQ was 1. So carbs. And how many hours after? Only after 1 hour, right? After 2 days, 48 hours, after 2 days, it's eating what? At 0.7. What is it eating? Fat. So after 2 days of not eating, all of a sudden it starts to eat fat. What does that tell you? It's eating its reserves. Does that make sense? It's not. It has no input, so it's eating what is fat used for then? To store energy. That's what we use our fat for. That's why organisms spend all summer getting fat. That's why you see little fat squirrels at the end of summer, and come the end of winter, they're skinny again. Do you ever notice that? They do that because what do they eat all winter? The foods the fat that they stored plus whatever food they saved, right? So when you're looking at, oh, I didn't know that could do that. That was weird. I've never seen it do that before. All right, so we're good with that? You should be able to read that. Can you do that? It's not hard, right?
No.
Taking this number, what will I do for you? I will make sure that you have this formula, so you don't even have to memorize the formula, and you'll have this table. You need to be able to fill this out and read this chart. I think it's easy. It should be two nice, easy questions, but it will be more complicated. Like I'll throw two lines on it or some kind of crazy thing. All right, let's go ahead. Let's move in, and always resubmission's fine as long as I know you've resubmitted it. I'll look at it. There's your 3 steps again. That was today's bell work again. Here it is: glycolysis, Krebs cycle, electron transport chain. Where's glycolysis happening? In the cytoplasm. That's here, look. Somebody asked me where the cytoplasm was. It's inside the cell, inside the membrane, but outside the mitochondria. All right, so I see it's almost time to leave. Look at these and get ready for tomorrow. Let's look at these questions. First of all, again, I want to emphasize that I'm not asking you to know how many electrons are formed here, here, or here. You should know that this is the most amount is formed here, the second most is here, the third is here. I do expect you to know glycolysis is happening in the cytoplasm, the Krebs cycle and the electron transport chain happen in the mitochondria. I expect you to know that. Describe precisely in which part of the cell the following take place. Well, I just told you. Provide a clear explanation of what is involved in each of the following processes. Substrate level phosphorylation, oxidative phosphorylation. So there's 2 things going on here. Substrate level phosphorylation is here in glycolysis. Glycolysis is substrate level phosphorylation. That's making ATP using a substrate and some enzymes. Oxidative phosphorylation is when we use oxygen. We use oxygen here in number 3. And we're going to go through and I'm going to explain it. There's some better pages as we move forward. But bottom line is oxidative phosphorylation happens a lot quicker. You're using a concentration gradient of hydrogen ions to make ATP using ATP synthase. Very similar to what we looked at and studied in photosynthesis. Okay? Yes? All right, we're going to go look at it in a minute. Let's go on. Well, that's right. Glycolysis
It's being broken down to that.
to pyruvate. That's right. So let's make sure we get this straight. Glycolysis, describe which part of the cell is taking, where is it, in which part is it taking place? Glycolysis is happening where? Krebs cycle is happening in the matrix of the mitochondria. You should know that. I just said it. It's in the chart right above it. Electron transport chain is a cristae. That's the inner folded membranes. Yeah? I can't hear you. I'm sorry, I didn't hear what she said.
She said, "Can she have a pencil?"
Oh, somebody has it? Yeah, okay. Substrate level phosphorylation, what is it? What is phosphorylation? Let me, what do you think we call it when you have remember that's adenosine and ribose sugar connected to, and then we take this, what is this thing called? Phosphate. What do you think we call it when we do this?
It's changed. It changed to a triphosphate.
Right? It does change to triphosphate, right? Adenosine triphosphate, you're right. What do you think we call the process of adding a phosphate? That's right, you got it. Somebody's paying attention. So phosphorylation. Phosphorylation. That's what when we add a phosphate to it, we call it phosphorylation. So if you're dealing with, what's the word substrate mean? This word here, substrate. Substrate level. But what does the word specifically, what does the word substrate mean? It goes back to our discussion on enzymes. Anybody remember that? You have this thing, let's call it an enzyme. Usually it's a protein, can be RNA, but it acts as a catalyst, right? It's an organic catalyst. Right? Speeding up the reaction by bringing the reactants together. What do we call the thing that it acts on? Like the hydrogen peroxide that we used in the example earlier. What is it? This is H2O2, and this is catalase. If this is an enzyme, what is this thing? You can call it a bunch of different things. You can call this the reactant as part of the reactant as part of the chemical reaction. But when you're connecting it to a substrate or an enzyme, we call it a substrate. The substrate is the thing that the enzyme is acting upon. Is this thing. Is that understood by everybody? I hope so. I hope you understand that because it's important. Okay. So the substrate is indeed this thing that's being acted upon. All right. So what do you think we mean by substrate level phosphorylation? It means you're using an enzyme. Like where? Like in a place called glycolysis. Because in glycolysis, what's happening? We're starting with glucose, and we have a chemical reaction, another chemical reaction, another chemical reaction, another chemical reaction. We split them off. We end up with two what? One there, we have another one over here, right? And then we end up using, here we end up using 2 ATPs. You should know this. I hope you know this, but you end up making 4 ATPs. So you make a net of 2 ATPs, right? This is done by what? Different enzymes at every step. It's reaction after reaction after reaction, but you end up. This is like somebody in their garage making a pair of tennis shoes. You can make a tennis shoes if you're good. You can make a lot in a short period of time, right? I mean, I don't know how easy it is to make tennis shoes. I've never made them, but you know, a shoemaker sitting in their garage can make a shoe. Maybe he can make 3 or 4 shoes or 10 shoes a day. Great. It's fantastic. But in the electron transport chain, and we're going to look at it in a little bit, you're taking all these electrons that are being brought to it by the carriers, right? NADH, FADH2, are taking all these electrons from all over the place, from glycolysis, from the Krebs cycle. All those electrons these electrons are getting carried over here, and they're going to go to a chain of different proteins in the membrane in the cristae of the mitochondria, and the electron's going to come in at different places depending on what it is. It's going to get transferred to one, then the other, then the other, And then finally, this molecule of oxygen gas is going to come in and take it and make what? Oxygen gas is going to take the electrons. It's going to take some hydrogen ions. There's a hydrogen ions out here as well. It's going to take hydrogen ions and oxygen and some electrons. And what are you going to make? What do you make in respiration? For about the 10th time, what do you make in cellular respiration? What's the product of cellular respiration? Carbon dioxide, but that's already done, right? That was done before it entered the Krebs cycle. We'll go over that in a second. We do water. That's right. We make water. That's where water's made. This is where water's made. That oxygen takes that last electron and makes water. Now, what's beautiful is that photosynthesis in the first light reaction takes that water, takes the electron, and makes oxygen. So they're literally exactly the opposite reactions. So beautiful symbiosis there, right? Very, very, it's a gorgeous little cycle. So, but the bottom line is, this is oxygen. What do you think we call this? And by the way, this allows, why is this important? Well, what happens if oxygen's not there to pick up the electron? What happens? So let's think about it. I'm going to play this, and I played it earlier, and you might, if you listen to this video at home, you might hear it being played twice. But have you ever seen a conveyor belt? So I guess we have one. And so this is a really cool little video. It's about a minute long here. And then we get to the commercial. So it's a cool little video. Look at it. You see these? These would be electrons. Let's go back. So this would be like electrons. They're coming down the conveyor belt, down the electron transport chain. Zoom, 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 they go. The electrons get, do you see that? Getting transferred from one place to the other. One carrier to the next. Back and forth, all over the place. And what would happen if nothing takes, these electrons here are going to get taken away by this machine. Do you see that? What happens if this never gets taken, these don't get taken away? They're going to build up. Eventually, what's going to happen? It's going to overflow. It's going to collapse. The whole thing is going to collapse. Right? So you need to keep taking those electrons away. They need to keep going away. So if there's no oxygen to take that electron, what happens to the whole process, the whole chain? It collapses. It's literally like a game of hot potato. Have you ever played a game of hot potato?
Yeah.
If I sat here and gave this young lady a hot potato and she kept passing it out, and I just kept handing her hot potatoes. They're hot, so she can't hold on to it for very long, so she passes it on. If there's no one at the end, if they have no place to put it, what's going to happen eventually? We're going to stop, or we're going to start dropping them. The process cannot keep going if you don't have somebody to take that electron away. That's that final electron acceptor. That's oxygen. The final one that takes that electron and makes water. Otherwise, the whole chain stops. If that whole chain stops, you don't produce any ATP. So, that's why this is so important. It also, you saw how that conveyor belt was able to produce that many boxes in the case of the movie, right? This thing produces a lot of ATP because it's a manufacturing line. You have a machine, an enzyme called Atp Synthase, and instead of using substrate level phosphorylation, using what kind of phosphorylation? Oxidative. Why do we call it oxidative? Because it uses oxygen. So it's oxidative phosphorylation. I never spelled it right. Okay? So what's the difference between oxidative phosphorylation and substrate level phosphorylation? Substrate level phosphorylation only produces a little bit of ATP. You have one glucose producing two pyruvates and only a little ATP. There's only a little phosphorylation happening here. And you're using some you're dephosphorylizing in order to get some more phosphorylation. You do get to make a profit of 2, right? But the point is that there's a lot of work here and only a little bit of output. The real energy comes from these guys, the electrons. That's really the juice of this, the energy that comes out of glucose is really here in these electrons. And what's beautiful is that if you remember photosynthesis, that's where we put the energy. That's where we put the light. Where do we put them? In the electrons. And where do the electrons come from, by the way, in photosynthesis? From water. They came from water. When we split water, we took the the plants. When the plants split water, they took the electrons and the hydrogen ions went flying out. Here, we're taking those hydrogen ions and that electron, and we're putting it back together again. The two processes mesh, they work together. Even though there's many, many enzymes and many different chemical reactions, there's still the two processes fit perfectly together. Yeah. Okay. Because tomorrow's Thursday, but it's one of the last days we have to really go through this study guide. I want to finish going through this study guide. Okay? All right, guys, have a great next slide. Let's take a look at these. This is a fantastic diagram. I promise you there's going to be some questions on the test on this diagram. Here you have, what process is this? What is going on here? This is cellular respiration, but specifically what part? Glucose is here, and you're making pyruvate glycolysis, the breaking up of glucose. Where does it happen? Well, right there it says it. Cytoplasm. Now here you have glucose coming in at the beginning. So if you're eating if you have glycogen stored, you have glucose stored in your liver in the form of glycogen, or starch if you're a plant, or even starch would come in here as glucose, and you start to follow the whole process. Because you're starting at glucose, you're going to make CO2. You're going to make a lot of CO2. You're going to make CO2 here. Look at it. Here you're making CO2. Do you see it? Yes or no? Yeah, there's CO2 being made. If you eat fat, some of it will come here through glycerol, but depending on the fat, but the fatty acids, the glycerol is going to come in here, rather. The fatty acids, though, are going to come in here. Did you produce carbon dioxide with the carbons and fatty acids? No, you did not. You skipped the carbon dioxide part. So you produced less carbon dioxide. Remember that RQ number with fats? Remember it was much smaller? Did you remember that RQ number? If not, go back and how come you don't have some of you don't have it open? Why not? Where is yours? Okay, look at it. Flip the page back. Look at the RQ value. Look at the table. Do you see the fat RQ value is much smaller? It's much smaller because you're not producing as much CO2. Do you see it? It's coming in after you make CO2. CO2 is made here when you make acetyl-CoA. Acetyl-CoA is made from fats directly. So you're going to if you eat fat, you're going to produce a lot less carbon dioxide. That's an interesting point. This explains the whys. Remember I told you a minute ago that I'd explain why the RQ value is smaller for fats? So anyways, each of these what are these? What is glycogen, fats, proteins? What are those things? Where do they come from? They're going into the catabolic pathways. Where do they come from? Where do you get proteins, fats, and carbohydrates?
Right?
That's what you get out of them. I'm asking you, where'd you get the fat, the protein, the carbohydrates?
Wait, I thought you
Food. You get enough food. You eat it.
I got a question.
Yeah.
So I thought you only get proteins and like fats and like
Right.
starch and all that stuff out of ATP.
There it is. I'm showing you. I'm telling you. Here's proteins going in. What do you turn proteins into? Amino acids. You break them up into amino acids. What happens to amino acids? They turn to what? Pyruvate. Now we go to pyruvate. Now you can make acetyl-CoA. Here you're going to produce CO2. Boom, boom, boom. But of course, amino acids, they're not going to produce a lot of CO2 because there aren't as many, right?
How do you pronounce
Well,
that?
depending on what? Acetyl-CoA? Acetyl-CoA enzyme A. The short is acetyl-CoA. I'm not really concerned about that you know it or not. Just so you know what to know. What do you have to know from this chart? Here's what you have to know. That in the cytoplasm, you do glycolysis, you're taking glucose and you're breaking down to pyruvate. You need to know that you can put glucose, fats, you can put carbohydrates, fats, proteins, all of them feed into this pathway. I do not care that you know where. I'm not going to ask you where in the pathway it goes in. Is that okay with you? I just need you to know that you can take all of these things, these things that we call food, and we can put it into this process to get energy out of them. We can get energy out of proteins, out of fats, and out of carbohydrates. Are we good? All right. Nucleic acids too, by the way. We can get some energy out of them as well. So the other thing you need to know from a chart like this from glycolysis is that you're producing pyruvate. Pyruvate's an important molecule. You should know that word. So make sure you know what pyruvate is, that it's going. And of course, pyruvate's lost almost immediately after it's made, because what does it turn into? As it goes into this, when it goes into the mitochondria, it turns into what? Carbon dioxide. And what do you do with that carbon dioxide? What do animals do with carbon dioxide? Animals consume carbon dioxide? What do you do with carbon dioxide? What? You let it out. You breathe it out. So when you ate that candy bar for lunch or those hot chips for lunch, that's coming in here. You came in here, assuming you worked it, you didn't turn into fat. Because if you didn't use it, you're going to turn into fat. But if you used it, it came in here, it got broken down into pyruvate, and then what'd you do with the carbon from that sugar? You breathed it out. You made CO2. You turned that chocolate bar into carbon dioxide, and you breathed it out. That's what your breath is. That's why you breathe. You're taking oxygen in so that you can use it in the electron transport chain, and you're breathing out carbon dioxide so you can get rid of your carbon. That's it. I hope that's clear. And that's why we can measure how fast this is going by how much carbon dioxide is being used up here, or put out here, and how much oxygen is being used up further down in the electron transport chain. Because what's the last this page is the perfect summary. If you study nothing else in this whole packet, studying this page will allow you to get close to an A if you understand everything that's on this page. Here is oxygen. Where are you using oxygen in this process? Where is this? Electron transport chain. So the electron transport chain is where you use the oxygen. It's the final electron acceptor. The final electron acceptor. I've said that I don't know, I want to say 20 times over the last couple weeks. You need to know that. It's the final electron acceptor. All right? That's what takes the electron and puts it and makes what? What are you making? As you take oxygen and you mix it with half an oxygen molecule and mix it with some electrons, some hydrogen, what are you going to make? Water. And that's why you were right. You can measure how much water is being made, and that could help you measure how much photosynthesis is occurring. Or not photosynthesis, sorry, cellular respiration. So, final electron acceptor is oxygen, and that's why you breathe. There it is. That's why fish need oxygen. That's why you need oxygen. The only reason we use oxygen is not to breathe. The reason you die if you don't get oxygen is because you can't make ATP. Why can't you make ATP? If you don't have any oxygen, there's nothing to take that electron. If nothing takes this electron, these electrons do what? They get backed up. Do you ever see an assembly line? Right? The conveyor belt comes by and somebody picks a piece up off the belt. Do you ever see that happen?
No.
Never saw that happen.
Say it again.
The conveyor belt.
No. They do it like factory.
It's in factories. You never seen it in a factory, really?
No.
Isn't it an emulsion?
Say that again.
Only Fidelity offers 4 0
Let me show you.
ratio index funds directly to investors. And now we have 0 account fees for brokerage accounts.
Allow me to show you real quick.
Zeros really
And
add
if you're watching this, if you're absent, look at it. You see how they're coming down? That's basically how an electron. That's it. That's the electron transport chain. Those are the electrons coming down those boxes. So what happened? You stop. If you're not picking them up off here, if they don't get picked up, what's going to happen to these boxes? They're going to back up, and the whole thing's going to do what? It's going to collapse. It's going to stop.
So what happens when you stop breathing for a long time and come back?
That they back up and you stop. And what happens is you don't make this. And if you don't make this, what's this? What is this? Oh, I'm sorry. I'm not. I forgot to switch it back. So if you what is this? If you can't make this, what is it? Energy. Can't make ATP. You can't make ATP. You don't do anything that you need to do. You're dead. That's what death is. You can't. Without this, you can't function.
So is that a reason you get out of breath?
You get out of breath because your body is signaling itself that it needs oxygen. Why do you need oxygen? Because you're not producing enough ATP. If you don't produce enough ATP, your cells say, yo, I need more ATP. It does it through signaling. They don't talk, right? They send little proteins out or hormones or signals of some kind, and they send them out to your brain. Your brain receives that message loud and clear. There's not enough oxygen. And if that is not enough oxygen, and this actually measures the oxygen level in your blood, by the way. That's how it does it. But that there's not enough oxygen in your blood, your brain says, breathe faster. Heart, pump faster. Because it's not just taking the oxygen into your lungs, right? How does the oxygen get from your lungs into your blood?
Wait, what?
Breathe it in. It's in my lungs. How does it go from my lungs to my blood?
Breathe out. Right?
Diffusion.
I don't know that.
Diffusion, the oxygen goes right through your membranes into the blood. Now it's in the blood. Oxygen in it gets stuck. It gets bonded to iron in your hemoglobin of your red blood cells. There it is. How do you get the oxygen from there in your lungs to your little toe on your bottom left foot? The bottom of your little toe on your left foot. How do you get it there? From your lungs.
I don't know.
Think you know the answer.
He said question.
I breathe it in. It's in my lungs. Diffusion allows the oxygen to go from in my lungs to my blood. Now it's in my blood. It gets stuck onto the hemoglobin in my red blood cells. Now they're there. How do I get it from my lungs in my blood, the blood in my lungs? How do I get it from there to my little toe?
Blood circulation.
Circulation. My heart pumps.
What is veins?
My heart pumps. And of course they travel through the veins and arteries.
Which one do arteries are the one that go to the heart?
Arteries are going away from the heart and veins to the heart. So the oxygen gets down there and then gets, again, through diffusion, high to low, it goes out to the cells. So that's how you get oxygen to everywhere in your body. So if your body says, hey, I don't have enough oxygen, first thing that happens, you start breathing faster. That's true. What else is going to start happening?
Heartbeat.
Heartbeat starts to increase. Faster and faster, heavier breathing, faster heart rate. Why? Because you need to get that oxygen to the cells so they can make ATP. It's immediate. It's absolute. You need to have this happen or you're dead. So your body makes it very much a priority. By the way, if you want to know the totals from cellular respiration, electron transport chain makes 34 ATP. Krebs cycle, only 2. Glycolysis, 2. You know what? I'm going to say, go ahead and know that. Go ahead and know that. 34 ATPs, electron transport chain, 2 from Krebs cycle, and 2 from ATP. Obviously, the most amount of ATP is made where? And This is an answer for a previous, let me be very clear. This is an answer for a previous question. This is called oxidative phosphorylation. Why is it oxidative? Because it's using what? Oxygen. The electron transport chain uses oxygen. And it happens that this is the reason that it does that is because it's literally an assembly line. So the other two, the substrate level phosphorylation is like a little old man working in a shoe shop making one shoe at a time. Oxidative phosphorylation is like Nike with their conveyor belts. They're making thousands of shoes at a time.
Thousands of people with Tyrese every second.
Do you see my point? One, oxidative phosphorylation is quick. It makes a lot. Where substrate level phosphorylation happens in the Krebs cycle and glycolysis happens very slowly. Do not get out of dress code. You're in dress code all period. Don't get out of dress code when you're on your way out. After you leave, that's between you and the principal. But in here, you're in dress code, and wait till you get out to get back out of dress code. All right, have a good day. We'll see you tomorrow. We should be able to finish out the respiration tomorrow. So what is chemiosmosis? There's the word chemiosmosis. It's the,
ATP is double.
that's right. It's the movement of protons, hydrogen ions, in the electron transport chain. Let me use a different. The movement of hydrogen ions in electron transport, right? You're taking NADH and FADH2. Is that good? Everybody gets that? That's what we call chemiosmosis. It's kind of confusing to me. You know, it's hard when you're teaching kids repeatedly that osmosis is the movement of water. And then you use this word chemiosmosis. Now, chemiosmosis is not the same as osmosis, right? But the word osmosis is here, right? It is used in it. So it might be a little confusing for you that osmosis is there. But we're going to go ahead and just accept that that's the definition of chemiosmosis. Now, this, you've seen it before in the light reactions. Isn't this what we did in the light reactions? We created a concentration gradient, right, with hydrogen ions. How did we create the concentration gradient? We used what? Electrons. And the electrons moving from one carrier to the other pull the hydrogen ions from what? Notice how this is light gray and this is dark. Why is that light gray and dark? From low to high. That's why this is a gradient, by the way. This, what's on this arrow, that's a color gradient, right? And so you go from low to high. And we've created a high here, high concentration here, and that allows this enzyme called ATP synthase, to do what? What is this enzyme? This is ACE, ends with the ASE, so you know that's an enzyme. And what's it doing? Synthesizing ATP. What does synthesizing mean? Make. It makes ATP. From what? ADP and inorganic phosphate. And this does it repeatedly, quickly, like a conveyor belt. It does it very quickly. But what does it need to do? For this to happen, you have to keep getting these hydrogens to go from low to high. Where's that energy coming from? The electrons. What if oxygen's not here to take that last electron? Then it doesn't move anymore. Hydrogen, and as soon as this runs out, you're done. You can't recharge the battery. This is a battery. And you can't recharge the battery unless you have electrons coming in, just like your phone, right? You can't recharge your phone battery without electrons. You call it electricity, right? I don't care if the electricity comes from a battery or from the wall. you can't recharge the phone without electrons. It's the same with this membrane, this system. In order for you to recharge this high concentration of hydrogen ions, you have to have electrons moving through. That's it. But here we don't have an outlet. Your cells don't have a little outlet. Your cells are using glucose to rip the electrons off glucose. They got the electrons from photosynthesis. Is that clear? Yeah. All right. You should be able to answer all these based on what I just said. I just described to you what hydrogen does, what oxygen does. I explained to you what the carbon atoms during respiration, where they go. I described to you glycolysis, Krebs cycle, electron transport chain, the total. There's also a little chart on the previous slide, right? So you should be able to answer this, all of this. And you should be able to describe how ATP is made, because I just did it. If you can answer all these questions, I can't imagine you'll get anything other than an A on Monday. So if you can answer these questions on your own, honestly and truthfully, based on what I've just taught you, you got it. You got cellular respiration. That's all you need to know for the test. Let's move on to the next slide. For photosynthesis, for cellular respiration. Now the anaerobic pathways, this is someone exercising. Here's someone exercising. Here's someone who's starving to death. Here's someone who's exercising. Here is someone making beer. I think it's beer. Is it beer? Yeah, brewing beer. We brew beer and wine by taking bacteria, yeast, and putting them in a vat and sealing them in an oxygen-deprived environment, so there's no oxygen in there, and giving them food, sugar. Where does the sugar come from in beer? Hops. They come from malt. It's a grass. All right. Where does the sugar come from when you do wine? Grapes. Where does the sugar come from when you brew sake? Sake is Japanese. Rice. What about mead? Ancient Viking drink. Honey. You might have known if you watch movies. They talk about it all the time. Mead. Whiskey. Where does the rye, like in rye bread? You ever hear rye whiskey?
Like rye bread?
Like rye bread. The rye is a plant, and it has to make sugar. Plants. All the sugars come from plants, right? But which plant defines which alcohol you make? And each one tastes different because each sugar comes from a different plant which has different qualities, right? But in all of them, you're using yeast in an anaerobic environment, no oxygen. So the yeast have food, but no oxygen. They can't do cellular respiration, so what are they going to do? Fermentation. What kind of fermentation? Lactic? They'll make alcohol fermentation, which then makes you what? It gives you alcohol. That's how you get, well, hopefully you don't drink, but that's the whole point. That's how we make beer. That's how we make wine. Say what? Absolutely. Alcohol is a poison. It's not we use it to sanitize our cuts. So it's a point. We drink it because people get drunk. They like getting drunk. Initially, it was used to kill the bacteria in the water supply in Europe and in other places, because water that you collect from a stream has a lot of pee and poop in it. And so mixing it with alcohol kills the bacteria so people don't get sick and die. So initially that's why they did it, but then they found out, hey, whatever. And so it actually became a cultural issue. So it's a culture. It's in many countries around the world. It's a cultural phenomenon, right? Alcohol becomes a cultural phenomenon. So we have whole factories making alcohol now, whole cultures around alcohol. So this alcohol is made though from yeast. That's why I always laugh when people tell me as they're eating their sandwich made of bread and drinking their beer, they tell me, oh, I would never eat mushrooms because that's a disgusting little creature. Makes them mushrooms are fungi. I'm like, fungi made that bread rise, and fungi made that alcohol you're drinking. What are you talking about? This alcohol is it time to go? Or why are people putting stuff away? No, I got time.
No,
Oh, I thought people were playing home. Keep me honest, that's fine. So alcohol fermentation makes ethanol. Ethanol is the kind of alcohol we drink. Now you can make other kinds of alcohols for the record, but ethanol is made from some type of glucose. Again, which glucose you get, where you get the glucose from, helps define what drink you get, right? So it depends on which drink. Do you want a rye? You want a whiskey, you use rye. You want sake, you use rice. You want mead, you use honey. You want beer, you use hops or malt. They call them malt liquor, right? So glucose, C6H12O6, makes pyruvate. But if there's no alcohol, if there's no oxygen, you can't do cellular respiration. Why? Because the whole chain backs up. We just talked about it, right? But your body doesn't want to die. It wants to try to live. It has to find another way of doing it. So the yeast don't want to die just because they don't have alcohol. So they try to find something else to do. So they take that pyruvate and they make ethanol. They make ethanol. Not us, they do. And that ethanol, or ethanol, and then ethanol then can take away carbon dioxide. They actually produce CO2 when they do this. You see that? CO2 leaves, and then you have a waste product. It's called ethanol. They get a little bit of electrons. They get a little bit of ATP, so they can keep the process going. Because see, they have to do something with this pyruvate. So by doing this process over and over again, they get a little ATP. You see it right there? A little bit of ATP, 2 ATPs. That keeps them going. We also get a little bit of ATP. There it is. But we have to do something with the pyruvate. What we do is we make lactic acid. We don't produce any CO2 there. You see it? No CO2. That's why you saw us produce very little CO2 whenever you're doing fermentation, anaerobic respiration. Producing a lot less CO2. Could someone close that door? Any questions about this process? Wait, I can't hear you because those people in the hallway. Go ahead.
You said before when they made beer that they don't take oxygen, but the thing said that they use oxygen, so when they use the CO2.
They don't use this CO2. They just spit it. They breathe it out just like we would. Is that what you're asking me?
No, I'm saying before when you were explaining how to make beer, you said that they keep it in containers that make sure there's no oxygen in it. Where do they get the oxygen from? The pyruvate?
They don't use it. Oh, for the CO2 you mean? Oh, I see. Because there's an oxygen atom in CO2. You're like, where is it? Okay, yeah, good question. Yeah, from the pyruvate. Excellent question. So, yeah, CO2 gets made. By the way, that's what makes bread rise. So you take bread and you make, which is made of what? Wheat, right? Which is made, what's the main component of wheat? Hopefully you know there's protein in it, yeah, but mostly it's what? Carbohydrates, right? It's not mostly yeast. My God, a giant yeast? That would be gross. No, it's made mostly of flour, right? Which is crushed wheat, right? You take wheat. In fact, you ever see a windmill? Oh my Lord. Room 374, Mr. Mendoza speaking. May I help you? Morningstar is not here. Jackson is not here. So I don't know if they're testing. Oh, she's here. They need you in the office, dear. Take your things with you. She's here. Jackson's here, but Morningstar's not here. All right. Thank you. All right, let's try again. So it's interesting, where were we? Oh, yeah, bread rises. So you take in the old windmills, the wind would turn the turbine and they would turn giant stones. And what would you throw in front of the stone as the stone turns around and around? It's a giant heavy stone. What? No, you throw the grasses. You throw the wheat. And what's the stone going to do to the wheat? It's going to crush it, and it's going to turn into what? Flour. And that's how you made flour. And you still make flour that way, just you don't use a windmill to do it. That's how we use electricity to push the crush it. But we crush it, we can make flour. And you take that flour, mix it maybe with some eggs, depending on what kind of bread you want, right? You take and you mix it up with whatever, and you add what? How do you get the bread to go up, though? How do you produce the because you ever look at bread, there's little tiny holes in it. Where'd the holes come from? Well, the answer is yeast. So you mix it in with a bunch of baker's yeast. What are the yeast going to do with all that flour, with all that sugar? Because it's sugar, right? That's basically what it is, carbohydrates. What's the yeast going to do? They're going to eat. They're going to eat. And what do they do when they eat? They produce CO2. They're doing cellular respiration. Some of them might do even fermentation, but even if they do fermentation, what are they doing? CO2. What's going to happen is they make more and more gas. What happens when you fill up a balloon? It expands. As it expands, it makes little holes. That's how you get bread, the holes in bread. So now you put it in the oven. By the way, they call that proofing. You let them eat. You let the yeast eat. They make CO2, the dough rises. Then you take that bread and you put it in the oven. What happens to the yeast? No, it's risen already. It went up. It rises a little bit afterwards too. The yeast die. But you're left with bread with holes in it. Maybe a little, you might have made a little bit of alcohol, but what happened to the alcohol in the oven? It burned away. So mostly bread is carbohydrates that have been cooked in the oven with little holes in it because of yeast. So if you want risen bread, not flat bread, you need to have yeast produce some CO2. So that's one industrial use, or two industrial uses of both the anaerobic pathway and the aerobic pathway, right? Okay. Photosynthesis. We already went through this, so this should be a quick review. And we have, I think, just enough time to do it. A couple minutes, a few minutes here, 4 minutes. There's everything we talked about, right? There's the granum, there's the thylakoid membranes. These are membranes, folded membranes, just like the mitochondria. We're going to come back to this later, probably in January, how similarly these two organelles, how similar these two organelles are. It's very important. Here's their sunlight. There's photosystem 2. Sunlight hits, it splits water, right? Electrons come off, oxygen gets released, the electrons get taken from what? NADPH. And where do they go? To the Calvin cycle. That's what I is, right? So you should be able to do this, right? You should be able to fill this out. You should be able to tell me what the different regions are, right? You got the stroma. What happens in the stroma? The Calvin cycle. What happens in the thylakoid membranes? The photosystems. We're using chemiosmosis to move the hydrogen ions across, making high and low concentration gradients. All that should be clearly described, hopefully. Here is an electron micrograph picture of the stroma. Again, this is the picture of them. This is the picture of the grana. This is our drawing of the grana. This is two-dimensional, very two-dimensional. This is kind of an attempt to make it three-dimensional, make it more visually acceptable. So here you can use the product of photosynthesis. You can produce disaccharides, more than one sugar. So fructose, right? Fructose and glucose. You can make sucrose, table sugar. You can make starch, the stuff in a potato. You can take it and make cellulose, the paper and the chairs you're sitting on, or most of you anyways. The monosaccharides, like glucose, you can make it. So that's what they do with this. And you can use those to make proteins and phospholipids, etc. So here's this light-dependent reactions labeled D, and light-independent reactions labeled I. This again are the light reactions, light-dependent reactions, and this is known as the Calvin cycle, or Calvin-Benson cycle, because those are the names of the guys that worked it out. You should be able to answer all these, right? We're good? Good summary. Even though most people understand the light reactions, I'm still going to go over them because I want to point out chemiosmosis and how chemiosmosis is used in photosynthesis, and then go back and look at how chemiosmosis is used in respiration. As you saw in the quiz, I expect you to know what chemiosmosis is, right? All right. Carbon dioxide is there, sunlight. So you see, it's the light-independent reactions, light-independent reactions that use the carbon dioxide. This is where what enzyme fixes carbon dioxide? Rubisco. All right. The light-dependent reactions make ATP, NADPH, and hydrogen, and all that goes into the Calvin cycle. Out of the Calvin cycle comes a triose phosphate. Triose, os is sugar, and tri means 3 carbon. It's specifically glyceraldehyde 3-phosphate, but whatever. That 3 carbon sugar can be made in any of these things. You can take that 3 carbon and build anything. It's the brick upon which all things are made in plants and therefore later in you. All right, so it's a sugar, but we focus on this one. Where is it? On this one called glucose, right? That's the one we focus on because the reason we focus on it, obviously, is that respiration uses glucose as the beginning of glycolysis, and that's why that's the beginning of the cellular respiration pathway, and why we focus on that when we discuss it. But you've got to remember that this sugar can make any of these, and I'm going to mention that on the test. I'm going to ask that question on the test. Do you know that it's a 3 carbon sugar that's made and eventually can be made into glucose, or it could be made into other things? Glucose can then be made into cellulose or starch, because remember, starch is a storage form of sugar, cellulose is a structural form of sugar. Disaccharides, which is sucrose, that's the stuff we eat, right? So you can make any of these, and you could they can be used to make proteins and nucleic acids, etc. Remember, plants make everything for themselves. We eat to get these things, but plants make it themselves. All right, so really this is a really great summary of C3 photosynthesis. All right, so this is important. This is a 3 carbon sugar, right? That's just said it again. A 3 carbon sugar, so we call this C3. Does that make sense? Yeah, so this is easy. This is C3 photosynthesis. We've gone over it. This is the easy one. This is C4 photosynthesis. I'm going to expect. Here's something else is coming up on the exam. All that's going to be on the exam, just so you know.
So this going to be on the Monday one too?
Yeah. The whole pack. That's why I gave you a study guide. I gave you a study guide. It's not time to go. Please don't put things away. You still got it. Don't I have a minute? Is it time to go? No, I still got a minute. Thank you. So visible light. This is your study guide. It is due today. I'm sorry, not due. It was due Tuesday. So it should have been done. So we'll finish photosynthesis tomorrow. You'll have your quiz on cellular respiration tomorrow. And if you have any questions, tomorrow's the day to ask. And you'll have your study guide for your final tomorrow. Good luck. All right, so pigments and light absorption. We've gone over this. I don't want to go over this again, but there it is. Just clearly, gamma rays, X-rays. Obviously, visible light is where photosynthesis is going to be happening. And what of these, which is the light that's not being used? Green. All right, green is not being used. You don't have to know the wavelength. I don't expect you to memorize that. That's ridiculous. It's not physics. If this was a physics class, I would. I'd expect you to know at what nanometers you see light. That's fair. But this is a biology class. So this is the absorption spectrum. We have there's something called an action spectrum for photosynthesis, and of course we have pigments. That's all background information. This is the money shot right here, okay? We have absorption spectra of photosynthetic pigments. What are the three pigments involved in photosynthesis? Chlorophyll B, carotenoids, and chlorophyll A. There's actually two types of chlorophyll at least, and of course there's the carotenoids, which are, all of them are just pigments, guys. It's no big deal. Yeah.
What is the name of the pigment that is used to, like for orange leaves or yellow?
For
Like in fall.
Corona, yeah, like
for what?
for like different color leaves, like orange or yellow. Cool. Just that one.
Oh, you mean the fall colors that come out on the leaves? Oh, I see. Well, what's interesting is with fall colors is that these pigments they break down at different rates. So let's say chlorophyll A, what is it absorbing? Let's see, it's absorbing up here near 700 nanometers. Hold on a second. So if red and orange get absorbed around 635, so they get absorbed around from here 635 to about 700. 700 is red, and 635 is orange. So around here, you see that? So what chlorophyll is going to absorb red and orange? This one, right? Now, if these pigments together absorb everything but green, right? Green is always going to be there. You're always going to see a green light. Now, get rid of this one, and all of a sudden, it's not absorbing red or orange, right? You absorb a lot less red or orange. Does that make sense? You probably absorb a little bit of orange, definitely no red. So now you're mixing red and green. Whatever color happens when you mix red and green color coming at your face, at your eyes, whatever that color is, that's the color you're going to see when chlorophyll A starts to break down. So maybe that's a purple. Maybe you see, if you get rid of orange, let's say this one breaks down, this is what chlorophyll B, right? Chlorophyll B is absorbing at around 450, and 450, 500, let's see, there's not much happening here, so you're probably dealing with, let me see. So if you start to, if chlorophyll B is absorbing most of the light here at around 450 to 500, that's mostly blue, right? But it's also absorbing orange. So now if you get rid of chlorophyll B, if it starts to break down and now it no longer absorbs blue or orange, and you mix blue, orange, and green, those lights coming at you, reflecting off the leaf coming at your face, then you get whatever color that is. If the carotenoids, which are absorbing here in this spectrum, which is around 400, now you see that these overlap. So wherever they overlap, you're going to get some absorption, even if it does break down, right? So if we look at the carotenoids here, this is chlorophyll A. Chlorophyll A is absorbing here at, 420, 430. So violet, it's absorbing the violet. The carotenoids here, carotenoids, what color do you think they're absorbing? They're absorbing everything from 450 to 550 or something like that. So you're pretty much getting, you're pretty much, the carotenoids are pretty much taking care of the yellow range, right? And so you get rid of this, get rid of the carotenoids, they break down. What color are the leaves going to turn? Yellow, because that's the one that's not being absorbed anymore. Remember, these are absorbing those colors. Does that make sense? Kind of? No? Yes? All right. Anyways, that's the answer to that question. That's not going to be on the test, really. I'm not gonna ask you to identify what color the leaves change when you get rid of this pigment. That's not really gonna be the point here. If you get rid of the, if you get rid of the other ones, here's carotenoids, they absorb, actually they don't absorb yellow. Carotenoids here, they say strongly absorbs blue, violet, and appear orange, yellow, or red. So there's so if you think about it, if you get rid of the carotenoids, you're going to see blue, you're going to see the one that normally would absorb. So if you get rid of the other chlorophylls, then what colors would you get? Orange, yellow, or red, right? Green, it's always going to be there. Green's always going to, right? Because it doesn't absorb, none of these absorb green. But carotenoids, if you get rid of if you get rid of the chlorophylls, A and B, and you're just left with the carotenoids, according to this, it does what? You're going to see what? You're going to see orange, yellow, or red. So these are the colors that you see in the fall, right? Does that make sense? But what would have to happen is you'd have to get rid of what? The chlorophylls. Because what's absorbing those colors? The chlorophylls. All right. So what happens is the leaf dies. These chlorophylls start to break down. And so you're left with the carotenoids, and that gives you that orange, yellow, and red color for the leaves. Is that fair? Okay, so in any case, the light is being absorbed, and here you have a graph that you've seen before. I showed it to you a month and a half ago, and you looked at this graph, and you see the different absorptions. You see where things are being absorbed, how much photosynthesis is occurring? A lot. When no light is being absorbed, what happens to photosynthesis? Or very little light, I should say, is being absorbed. What happens to photosynthesis? It drops tremendously, right? So there's a big difference between the least and the most here. Here again, you're absorbing light again over here, so again, you're doing photosynthesis over here. So you're getting a lot more photosynthesis where you have a lot more absorption. Is that fair? All right. So you should be able to answer that. This is a graph that you should be able to read. Expect to see it. I've seen it on practice OST questions. I've seen these on standardized exams ranging from ACTs, AP exams, AP biology exams. You're going to see stuff like this. It's going to be over again. That's a classic chart. Here's photosynthetic ability. So as light intensity increases, right? As light intensity increases from 1 to 7 lumens, it's some kind of scale, whatever it is, your rate of photosynthesis does what? Increase. And it looks like they're measuring rate of photosynthesis by how much CO2 is being used, right? So you see it increases as you increase light. That makes sense. Light intensity is CO2 and temperature versus photosynthetic rate. So as light intensity goes up, photosynthesis goes up, but look at the different temperatures. Look at what happens at low temperatures. Right? So here at high CO2 at 20 degrees, here's low CO2 at 20 degrees, here's high CO2 at 30 degrees. Look at the difference in temperatures here. Even though you have a lot of CO2, you have less photosynthesis. So the colder you get, what? Less photosynthesis is occurring, even when you have plenty of CO2. So this is showing you the relationship between temperature, light intensity, and how much photosynthesis is occurring. You should be able to answer those questions. I'm not going to worry, but these are the kinds of charts you're going to see on the test on Monday. So get to, be able to read them, be able to answer questions like these here. Of course, your questions on Monday will be multiple choice. All right, so let's take a look at it. This is the light reactions. It's pretty straightforward. I love this diagram much better than the one I drew for you in class. I was hoping you guys would take the time and do it, and you certainly, some of you did. Chlorophyll 2, all right, chlorophyll. Of course, the carotenoids are involved here as well, all right? So they're absorbing light. They're taking these lights in. This is all obviously very simplified. Photosystem 2, what's the first thing that happens? Right? Light splits water. This is the question I asked you today. I asked you, what's the connection between photosynthesis and respiration? Somebody wrote something very good. They didn't get any points for it, but I like what they did. What they wrote, because it meant that they paid attention, but it also meant that they didn't do enough work to get their answer right. The part that showed me they paid attention is that the products of one feed into the other. When I asked you about the relationship between photosynthesis and respiration, that's true. That is true. The products of photosynthesis feed into respiration. The products of respiration feed into photosynthesis. That's right. What products? So water feeds into photosynthesis. Where's water made? Cellular respiration. Oxygen leaves photosynthesis. Where does oxygen go? Cellular respiration. So you're right. The person who wrote that, what's made in one is used by the other. You're absolutely right. I'm glad you remember us saying that in class, but you need to learn exactly what. So you're passing the electrons. It doesn't. I don't like this that they don't show light coming and hitting this electron, because light does hit this. It just doesn't show it here. The electron's high energy because of this light. It drags the hydrogen ions from low concentration to high. This is the sort. This is chemiosmosis, by the way. Does everybody understand that? This hydrogen ions going this way and making ATP. That's chemiosmosis. This whole process. Do we understand that? Yeah.
So, could an answer to that have been like turning light energy into chemical?
No, unfortunately, that wouldn't be. That's the light reactions. The answer to that would be anything that shows that you understand it's the electrons being used to move hydrogen across the membrane, and then hydrogen ions moving from high to low to make ATP. Anything that's close to that is going to get you the right answer on that question. Yeah.
Would it have been acceptable if you would have said the diffusion of hydrogen ions over a semipermeable membrane and to make ATP all over?
Yeah, I think that's okay. I wish you would have mentioned something about the electrons, right?
Oh yeah, I said they were getting pulled down because of the electrons.
Yeah, then that's right. That would be that's the right answer. Anything like that shows me you understand the concept, because the whole point is that this is what happens in the light reactions. Where else does this happen? Where else does this happen? Cellular respiration. What part of cellular respiration does this happen in? Mitochondria. What process? Electron transport chain. Electrons being transported. So this is very similar to the electron transport chain in cellular respiration. The process is conserved. Once life finds something that works, it sticks with it. It works until it doesn't, until something better comes along. That's how we do. That's how everything does. From bacteria to trees to humans. If it's working, fine. When it's not working, you die, you don't have babies, and so your genes don't get passed to the next generation, but the ones that work make babies and their genes continue on. And if somebody else works better, their genes get carried on more often than the person who has the less functional one. So eventually, over time, the ones that work the best win. Does that make sense? So it's a kind of a very basic, even at the molecular level, this is a kind of an application of evolution. So photosynthesis 2, photosynthesis 1, photosystem 2, photosystem 1. I think most of you understand it based on some of the previous assessments, so I think we're good with that. Now the Calvin cycle, people are confused about it. Now, I warned you that there's a lot of steps here, right? There's enzymes, there's substrates. You don't have to know all these steps. I like that simplified version we'd gone over in class. But here, 3 molecules of, first thing that happens is that the pyruvate goes into the mitochondria. Remember the pyruvate? Where do we make pyruvate? Oh, I'm sorry, this is a Calvin cycle. My bad. I got confused there for a second. CO2's coming in. What's fixing CO2? Glucose is not fixing CO2. Fixing CO2. Fixing means capturing. What's capturing that CO2 and holding it? Rubisco. And there it is. Let me highlight it for you. There it is. Rubisco. Oh, that's not good. It doesn't show up very well. Let me do this. Rubisco, all right? Rubisco is the enzyme that captures CO2 and brings it and allows the Calvin cycle to do what? Make sugar. That's what the Calvin cycle is going to do. It's going to make the sugar. It needs to put that energy that photosystem 2 and photosystem 1 captured from the sun. You need to put that energy into the chemical bonds that make up sugar. That's why sugar has so much energy. Because the plants put the energy from the sun into sugar. So it takes the CO2 from the air. Takes it in from the air. So there you go. And do you need to know any of these things? No. Do you really need to know how many ATPs go in, how many ADPs go out? No. In AP biology, you might have to learn that. In college, you'll certainly have to learn that. In college, you'll have to learn what the names of each of the substrates are and the enzymes. That's fine. But here, the only thing you need to know is Rubisco, CO2's coming in, being captured, and then out comes glyceraldehyde 3-phosphate. Then the rest of these components get remade. They just keep getting remade. That's it. So the only thing you have to know is what? NADPH comes in. Where'd the NADPH come from? What's it bringing? Electrons to the Calvin cycle. There's electron carriers. It's bringing the energy. What are the electrons carrying? The energy that it captured from the light. Wow, it's 8:59 already? Dang, the whole day went.